This video demonstrates a protocol for isolation and phenotypic characterization of hematopoietic progenitors using magnetic bead-based separation and fluorescence-activated cell sorting. First, the hind leg femur, tibia, and spinal cord are dissected from a mouse and homogenized to generate a bone marrow cell suspension. The population is then enriched for cells that express c-KIT using magnetic microbeads. Flow cytometric analysis of the enriched population demonstrates that this method yields highly purified hematopoietic lineage progenitors and hematopoietic stem cells, which can then be used for in vitro and in vivo functional studies.
The main advantage of this technique over existing methods like bone flushing is that it allows more efficient and faster recovery of bone marrow cells. The implications of this technique extend toward the therapy or diagnosis of inflammation, autoimmunity, immunodeficiencies, degenerative disease, metabolic disorders, and cancer in murine models, because it allows the phenotypic characterization and ex vivo functional analysis of hematopoietic progenitors. So this method can provide insight into monitoring hematopoiesis in different physiological and pathological conditions. It can also be used to study the cell biology, signal transduction, and developmentally regulated gene expression in distinct progenitor subsets.
Begin by placing a euthanized mouse in a stainless steel pan. Spray the abdomen and back with 70% ethanol. To collect the bones that will be used as the source of cells, use scissors to make 2 small openings in the skin at the hind legs. With the help of forceps, dislocate the femur. Then, turn the mouse upside down and cut the skin longitudinally to display the spinal column. Remove the spinal column. Then, using sharp-tipped scissors, carefully remove all soft tissue residues from the spinal column. Use gauze to remove soft tissue residue from the femur and tibia. Then, place the bones in a 50 ml conical tube containing 30 ml of supplemented RPMI medium. Place several 4/5 sterile filters in a sterile mortar. Then, using forceps, transfer the bones into the mortar and cover them with another layer of filters to assemble a friction surface. Next, place a 40 micron cell strainer filter in a 50 ml conical tube. Then, using a pestle, smash the bones until a homogeneous cell suspension is obtained, and, using a pipette, transfer the suspension into the cell filter. Next, add 5 ml of fresh supplemented RPMI to the mortar to wash and harvest the remaining cells. Pipette up and down. Then, add the RPMI to the tube with the filter. Repeat the last 2 points until the homogenized bones appear clear. Once the solution has passed through the filter, transfer it to a new 50 ml tube, and filter, and repeat the process to eliminate the remaining tissue debris. Centrifuge the tube at 500 times G for 5 minutes at 4 degrees Celsius, and carefully remove the supernatant by aspiration. Make sure not to disturb the cell pellet. The next step of the procedure is to characterize the phenotypes of hematopoietic progenitors. Begin by resuspending the cell pellet from each mouse in 15 ml of PBS and 2% FBS. Then centrifuge the tube at 500 times G for 5 minutes at 4 degrees Celsius, and carefully remove the supernatant by aspiration. Do not disturb the pellet. Resuspend the pellet in 5 ml of PBS and 2% FBS and count the cells using a hemocytometer. Once the cell concentration has been determined, transfer 1 × 10⁶ cells to each well of a 96 well round bottom plate. Then, centrifuge the plate at 500 times G for 5 minutes. During the spin, prepare a mix of monoclonal antibodies directed against the surface markers listed here in PBS and 2% FBS to stain the cells. Once the spin has completed, discard the supernatant by inverting the plate and add 50 microliters of the antibody mix to each well. Then, using a multi-channel pipetter, pipette up and down to dissociate into single cell suspensions. Incubate the cells for 20 minutes in the dark at 4 degrees Celsius. Following the incubation, wash cells twice with 150 microliters of PBS and 2% FBS. Then, centrifuge the plate at 500 times G for 5 minutes. Then, discard the supernatant by inverting the plate. Next, add 50 microliters of diluted 1 to 300 AP3 size 7 streptavidin in PBS with 2% FBS to each well. Dissociate into single-cell suspension by pipetting. Incubate the cells for 10 minutes in the dark at 4 degrees Celsius. Following the incubation, wash the cells twice with 150 microliters of PBS and 2% FBS. After centrifuging the plate again, discard the supernatant and resuspend the cells in 100 microliters of PBS and 2% FBS. Analyze the samples by flow cytometry. Next, to sort selected subsets of hematopoietic cells, magnetic enrichment is performed on bone marrow to enrich c-KIT positive cells. Resuspend the cell pellet of bone marrows derived from at least 10 mice in 1 ml of buffer solution containing c-KIT monoclonal antibody conjugated to APC, diluted 1 to 50. Incubate the cells for 20 minutes in the dark at 4 degrees Celsius. Following the incubation, wash the cells twice with 40 ml of buffer solution. After centrifuging, carefully remove the supernatant by aspiration. Then, add 50 microliters of anti-APC microbeads per mouse directly to the pellet. Mix well and incubate for 20 minutes in the dark at 4 degrees Celsius. Following the incubation, wash and spin the cells as before. Then, resuspend the cell pellet in 4 ml of buffer solution for every 3 mice. Place max LS columns in the magnetic field of a suitable max separator. Prepare the columns by rinsing with 4 ml of buffer solution. Then, apply cell suspension and collect the unlabeled cells that pass through in a collection tube. Wash the column with 4 ml of buffer solution 3 times. Add new buffer when the column reservoir is emptied. Once all of the solution has flowed through, discard flow through. Remove the columns from the separator and place each one on top of a 15 ml conical tube. Pipette 4 ml of buffer solution onto each column. Immediately flush out the magnetically labeled cells by pushing the plunger into the column. Centrifuge the tubes at 500 times G for 5 minutes. Carefully remove the supernatant by aspiration. Resuspend each pellet with 1 ml of buffer solution and group pellets in a single 15 ml conical tube. Once the c-KIT positive cell population has been enriched, stain and sort the cells by fax. Hematopoietic progenitors and hematopoietic stem cells from a bone marrow suspension enriched for c-KIT positive cells using magnetic beads and LT-HSC and ST-HSC were sorted according to expression of surface molecules as described in this video. This figure shows an example of the electronic gating procedure to score common lymphoid progenitors as Lin-c-KIT low interleukin 7R positive cells, Lin-c-KIT positive SCAR1 positive, and Lin-c-KIT positive SCAR1 low cells. From the c-KIT positive gate, common myeloid progenitors are identified as Fc gamma R low CD34 positive cells. Also from the c-KIT positive gate, granulocyte monocyte progenitors are identified as Fc gamma R high CD34 positive cells, and megakaryocyte erythrocyte progenitors are identified as Fc gamma R low CD34 negative cells. From the LKS gate, LTHSC and STHSC are identified as CD34 negative and CD34 positive cells respectively. Thus, 2 enriched and highly purified populations are obtained that are physiologically present in low number. To address the impact of autoimmunity on hematopoiesis, HSCs from control and mice with inflammatory bowel disease were stained with Ki-67 antibody and DAPI. As can be seen here, cycling cells are barely, if at all detectable, in the LKS CD34 negative hematopoietic stem cells compartment of healthy controls. In contrast, the same cell subsets is significantly increased during IBD, and a substantial number of HSCs are detected in the S, G2, M phases of the cell cycle, thus suggesting that T-cell-mediated chronic inflammation determines entry of HSCs into the cell cycle. To assess the presence of ATP stored in vesicles, CD34 negative cells from healthy mice were stained with the nucleotide-binding compound quinacrine and nuclear red. Analysis by live cell confocal microscopy reveals that in HSCs, but not GMPs, quinacrine-positive vesicles are visible in the cytoplasm. To determine whether HSCs are responsive to extracellular ATP, the calcium response of sorted hematopoietic stem cells loaded with FURA2 and stimulated with ATP and ionomycin was analyzed. As seen here, single-cell calcium elevation traces indicate that there was an increase in cytosolic calcium in LT-HSC by activation of purinergic P2 receptors upon exposure to ATP. Since cytosolic calcium elevations induce release of vesicular ATP, this experiment suggests that ATP may regulate its own release from HSCs and demonstrates that this procedure can be used for functional studies of HSCs.
After watching this video, you should have a good understanding of how to isolate murine hematopoietic stem cells and lineage-committed progenitors for functional assays. Following this procedure, other methods like reconstitution of non-irradiated recipient mice can be performed in order to answer additional questions, like the reconstitution potential of hematopoietic stem cells purified from steady state or inflammatory environment.